All right, well, welcome back, guys. This is lesson 3 in our Intro to Genetics series. We are applying Mendel's principles specifically this time to what we would call a dihybrid Punnett square. So a dihybrid cross was simply, di means two. We're literally just going to look at two different traits at the same time. Now, that's actually what is shown to you right over here in this picture. So we're going to dive into that and break it down. How do we set up such a cross? Okay, so here we have two individuals. These are pea plants from Mendel's pea plants. I've just made them purple and green just so we can easily see them and designate which is which. What we have to do first, you're going to notice it's a little bit different. There are a lot more squares in this Punnett square, which again is not a square. It's actually more of a rectangle. But anyways, it's got 16 boxes. It's a dihybrid cross. I'm going to show you two different ways to do this. First, the kind of traditional way of actually filling out the big Punnett square, and then I'm going to show you a much faster way where we multiply some ratios. We'll break it down into two smaller Punnett squares and we'll multiply some ratios. So first, here is the kind of longer, more tedious way, but you need to see it. So what we're going to do is we're going to basically do FOIL. So again, we're going to relate this back to math class. We're basically going to FOIL this, and what we're going to do is I'm going to take the first of the two letters in the two genotypes that are side by side for this individual, and I'm going to write them here. So in this case, we have a big R and a big Y. They'll go here. Next, I'm going to do the outer ones, big R, little y. Now I'll do the inner ones, little r, big Y. And the last one's little r, little y. And then I will turn right around and I'll do the same thing over here. FOIL it first. And you're going to notice these are going to look very similar. Outer, because they have the same genotype. Inner, and last. Okay, now I'll simply fill these in the exact same way that I normally would. All right, so let's go with, let's fill these in with white. Actually, no, let's switch colors. Let's do something a little brighter so it stands out. All right, so I'm going to take this and combine them right here in this box. So I'm going to have two capital R's and two capital Y's, and I'll just fill in the Punnett square the exact same way. So this is really not that hard, filling in the Punnett square. So again, I'll take this, now this, big Y, little y. That's not the hard part. What I typically lose people on, what typically gets them in some trouble is when it comes to setting it up, making sure that they get the genotypes correct. So if you're working in your workbook right now, my Scott High students, this specifically, you'll find this on page 146 in chapter 12 of your biology workbook. This is the exact example that is shown there. And so I'm going to show you how to get the results and the frequencies here in just a moment. But the setup is initially what confuses most people. So let's finish filling this in really quick. So now what we have here is we have the dihybrid cross complete. And if you're working in your workbook on page 146, it wants you to identify the genotype frequencies. All that means is go through and count the combinations and see how many times that particular combination shows up. So I'm going to actually type those in because my handwriting will not possibly fit in here. So R, big Y, and you end up with 1 out of the 16 that has that. Okay, so same thing here. Then our next one, okay, and so I'm going to cross that off the list as we go. All right, so I have completed that one. Next, I see big R, big Y, little y. Okay, so I see two here. Here's one, and that is it. So then I'll go to the next one. Heterozygous for the R's, 2 capital Y's. I see 1, 2. That is all I see for that. Okay, so this is quite tedious. I'm actually not going to finish this out because that just gets really tedious. You get the idea. I'll allow you to finish that. What I want you to do, really focus on though, is at the end of this, what you will end up with. I'm far more interested in the phenotypes for this particular cross. And what we would do to figure out how many are going to be round yellow, we will need to go find any that have a capital R and a capital Y. So here is 1 with a capital R, capital Y, 2 with a capital R, capital Y, 3, 4, 5, 6, 7, 8, 9. So there are 9 out of 16 round yellow. So a 9 out of 16 probability of having round yellow seeds. What about round green? So that would be a capital R and 2 lowercase y's. I count 1, 2, 3 that have a capital R and 2 lowercase y's. So that is 3 out of 16. If this is a little too quick for you, which leaves us, if you do the math, we only have 1 left. So only 1 out of the 16 will possibly have wrinkled green seeds. And so that is a dihybrid cross. Now that is the difficult method, the difficult way of doing that. I would like to show you a much easier way. All right, so what I'm going to do now, we're going to go with the same genotypes that we started with, okay? But what I'm going to do is I'm going to break them down into a much easier method of figuring all this out. All right, so obviously we already know what these are going to end up being. So I'm just going to leave these here. Here are our final results that we know that they're going to turn out to be. But here's what I'm going to do. I'm going to get rid of this. And very quickly, here's what I will do. I'm going to break this down into 2 Punnett squares. Okay? It works, trust me. So what I'm going to do is I'm going to look at the R's. All right, and I'm going to construct 1 Punnett square just for this trait. And I'm going to figure out the ratios for it. All right, and so I end up with 3/4 round and 1/4 wrinkled. Now I will do the same thing for the Y's. I will construct a quick Punnett square. And I'm actually going to find that it's the exact same ratios. All right, but this time it's yellow and green. Okay, now what I need to do in order to figure out my results over here for each particular phenotype, in this case, let's do the first one, round yellow. I'll simply take the number for round and the number for yellow and multiply them together. So 3/4 times 3/4. Which gives me 9/16 for round yellow. And is that not what we found up here? We got 9/16. Now, it works the same way. So let me erase this. It works the same for all the others real quick, and then we'll wrap up this video. Round green, take the number for round, the number for green, 3/4 times 1/4. (3/16). That is correct. Same thing for wrinkled yellow. 1/4 times 3/4 is also going to be 3/16. That's what we found it to be. And last but not least, wrinkled green. 1/4 times 1/4 is going to be 1/16. And that is indeed what we found. So this is actually a much faster way to do it. I prefer this method. If it confuses you, you can always do the bigger 16 box Punnett square, but I prefer to multiply the ratios. And so I hope this has helped you complete lesson 2 in your workbook. And if you're not one of my students at Scott High and you're just trying to learn along right alongside with us, I hope this has helped you learn how to do dihybrid Punnett squares. As always, I want to encourage you to subscribe to my channel, like the videos, comment, leave me comments. I'll get back to you on that. And watch out for lesson 4 in our Intro to Genetics series.